Hello, in this video I will talk about an interesting biochemical test that are performed on microbes to categorize them. The name of this test is Methyl-resand voges-proskaur test, MRVP test in short. Both these tests are taken simultaneously and are thus performed on the same medium, MRVP broth. It is not a solid medium in which it is performed, so it is taken as broth, MRVP broth. Now a question that will come to our mind, that why these tests are performed simultaneously. This is because a single isolate or culture of microorganism will show opposite results and are usually obtained for the Methyl-resand and voges-proskaur test. That is, the organism will either show MR positive result, VP negative result, or MR negative result, VP positive result. This test is used to differentiate two major types of facultatively anaerobic enteric bacteria that produce large amount of acids and those that produce acetone as end product. So the organisms that will form mixed acids are to be called as MR positive, and those that will produce acetone as end product will be called as VP positive. Now let me give you a brief introduction to this test. During glucose fermentation, we all know the end result of fermentation are acids and alcohol. Carbon dioxide is also there. So during glucose fermentation by this anaerobic enteric bacteria, that results in end products and which ultimately determines, means the fermentation is dependent on the metabolic pathways that are available to the microorganisms under the culture condition. A number of genera of bacteria ferment glucose to produce lactic acid, acetic acid, succinic acid, and formic acid, that is mixed acid fermentation. They also produce carbon dioxide, hydrogen, and ethanol. These acids produced can reduce the pH to 5 or lower, while other organisms ferment glucose to form only one product, usually acetic acid, which is later converted to acetone. So when the pH drops below 5 or 4 around, then it is called as MR positive because the acids have been formed. And when the pH does not change or increases, means acetone is produced, and it means VP positive result. In MR test, when acids were produced, the pH drops below normal. Methyl-resand, an indicator, turns red when the pH comes below 4.4. Otherwise, the indicator will remain yellow. So if it is VP positive, MR positive result, the indicator will turn to red. Otherwise, it will remain as yellow. In VP portion, when the reagents are added, it will react to acetone and show a red color. So this is a short introduction to this test. Methyl-resand and the voges-proskaur tests are used to differentiate two major types of facultatively anaerobic enteric bacteria that produce large amount of acids and those that produce acetone as end product. These tests are performed simultaneously and are performed on the same medium, MRVP broth. Opposite results are obtained for the methyl-resand and voges-proskaur test, that is MR positive VP negative or MR negative VP positive. Principle: in these tests, if an organism produces large amount of organic acids like formic acid, acetic acid, lactic acid and succinic acid as end products from glucose, the medium will remain red, which is a positive test. After the addition of methyl-resand, a pH indicator, that is pH remaining below 4.4. In other organisms, methyl-resand will turn yellow, a negative test due to the elevation of pH above 6 because of the enzymatic conversion of the organic acids produced during glucose fermentation to non-acidic end product such as ethanol and acetone. MRVP broth tubes were inoculated with the isolate. So after 48 hours of incubation, to 1 drop 5 drops of methyl-resand indicator was added. To the second tube, 12 drops of VP reagent 1 and 2 to 3 drops of VP reagent 2 were added, and one tube was kept as an uninoculated comparative control. Materials required: preparation of the MRVP broth maintaining pH 6.9, tubes of the following composition: peptone, dextrose, glucose, potassium phosphate and distilled water. Procedure: pour the 5 ml broth in each tube and sterilize by autoclaving at 15 lb pressure for 15 minutes. Inoculate 2 MRVP tubes with isolate and keep one tube as uninoculated comparative control. Incubate all tubes at 35 degrees centigrade for 48 hours. After incubation, 5 drops of methyl-resand indicator to be added for each tube, each set. Observation: observe the change in color of methyl-resand for MR test. Add 12 drops of MR reagent 1 and 2 to 3 drops of VP reagent 2 to the other set of the tubes as well as to the uninoculated control tube. Shake the tubes gently for 30 seconds with the caps off to expose the media to oxygen. Allow the reaction to complete for 15 to 30 minutes. Observe the change in color for the VP test.